All right, so you're going to find that all of our units in this course kind of follow the same pattern. Okay? In the chemistry unit, the first day we started talking about the basic unit of chemistry, the atom. Well, today we're going to start biology, and we're going to talk about the basic unit of biology, the cell. Okay? And we're going to talk a bit about the history of the cell, stuff like that, just like we did with chemistry. The good news is there aren't nearly as many people involved for this one. All right, we had to learn all, Thompson and Dalton and all those other guys. Okay, this one's like 4 people. Okay, that's it. All right, and 2 of them work together, so it's really like only 3. So we're going to be looking at that and then talking a bit about the microscope and how the microscope works, so that we're kind of ready to go for tomorrow's lab. Tomorrow's lab is not really going to be anything earth-shattering. It's more about becoming familiar with using the microscope, understanding how the microscope works. Okay, so it's not really going to be about like labeling the parts of a cell. Certainly we're going to do a lab drawing, okay, or lab diagram, because you're not actually going to draw it. They actually use Google Draw to do it. It's much easier. You just use take a picture of it with your phone and then label the picture. Okay, but yeah, we're going to be looking at stuff like that, just kind of getting used to using the microscope. And then our next lab, which will be Monday of next week, we'll be looking at actually labeling parts of a cell, because we'll be more familiar with what everything looks like and using the microscope by then.
Are we going to have our quiz tomorrow off what we learned today?
Possibly. See how far we get. All right, so for today, we need to know the points of the cell theory and how they relate to living organisms. Okay, so there's only 3 points to the cell theory. There were 4 in the atomic theory, so you're getting a break already. Okay, only 3 points. Know who contributed to the development of the cell theory. Like I said, there's literally only 4 important people. Okay, and recognize the implication of the similarities between plant and animal cells in terms of evolutionary relationships. Okay, so what I mean by that is, this is a plant cell. Okay, but if I put an animal cell beside it, they actually have more in common than they have different. Okay, and what that says from an evolutionary perspective is that plants and animals had a common ancestor at some point, okay, in history, and that ancestor possessed all of the structures that they still share. Okay, there's virtually no chance of 2 completely different organisms developing cells that are so similar randomly. Okay, like completely separate. There's basically no chance of that. Okay, and when we trace the fossil record back, that is what we see. Okay, the first living organisms were single-cell organisms that had similar functions, and then through evolution divided apart. Okay, like broke off into plants, animals, fungi, bacteria, stuff like that. All right, so that's what we'll be looking at as well. Okay, first point of the cell theory. Okay, actually, let's go back one here. Let's brainstorm. Okay. What are some characteristics of all living things? Okay, yes.
Yeah.
They use oxygen.
Okay, they have some kind of metabolism. Okay, use oxygen. Okay, what else? Okay, made up of cells. Because I showed you guys the first point, this is going to ruin my demonstration, but okay. Because this is usually the one people miss. Okay, usually people are like, yeah, they use oxygen, they produce waste, they reproduce, blah, blah, they talk about all these things, and then I say to people, so fire is alive. Right? Because usually they miss this part about being made of cells. Okay, and then I say, so fire is alive, right? Because fire uses oxygen, it produces waste, it consumes fuel, okay? It can reproduce, one spark can start another fire, okay? No different than binary fission for a cell, right? Stuff like that. If you look at it, that's usually what people list, and all of those things can fit with fire, which is not a living thing. It acts like one sometimes, okay? But it's obviously a chemical reaction and not a living thing, right? This is the thing that qualifies something as being alive. Now, we say that to be alive, something has to be made of cells. But viruses are not made of cells. Okay? Are viruses alive? Are they a living thing? There's a lot of argument, actually. The debate rages, to be perfectly honest. The debate rages as to whether a virus is a true living organism. Because all of those things that even fire can do, viruses don't. Viruses do not consume food. They do not produce waste. They cannot reproduce by themselves. Okay? They only reproduce by hijacking your cell and making it produce more viruses. Okay? So the virus itself does not actually reproduce. Okay? It uses something else to make more copies of it. Right? So it actually doesn't fit any of the stuff that we would have listed were characteristics of a living thing. Okay? Really, the only thing it has in common with other living things is genetic material. Right? You guys know how a virus works? You've been taught that? Sort of? Okay, so what a virus does is it will latch on, okay, to your cell. So if I'm drawing like a bacteriophage, they kind of look like this. They almost look like the lunar lander, okay? And they have these things that kind of grab onto the cell membrane. And then inside the top part, and with any virus, there's just a shell, a protein shell that contains the genetic information, okay? The genetic information is then injected basically into the cell. Cell's natural reaction to the presence of genetic information floating freely inside the cell is to bring it into the nucleus where all the genetic information is kept. And at that point, it gets written in to your cell's DNA. Okay? So basically, a part of your cell's DNA gets cut out, just that cell, okay, gets cut out, and the virus's DNA gets spliced in its place. Now, what your DNA does for you is it tells your body how to make stuff. So if you need to make, let's say, insulin, for example, part of your DNA, some piece of the strand, is the genetic information and the instructions for making insulin. If the virus has spliced itself into that section, the next time that cell needs to make insulin, it doesn't make insulin, it makes a virus. Okay? Now this starts to feed back because the cell needed insulin and didn't get it. So it sends the request again. Need insulin. Nucleus goes to the section of the DNA that makes insulin, goes, all right, there we go, make that. Makes another virus. And it just keeps going, okay? Pretty soon the cell is full of viruses, okay? And it's starting to malfunction because it's not making the stuff it needs anymore, but it's making all these viruses. Eventually the cell will rupture and all those viruses will get out, okay? And then they will go and infect other cells. Now, this could be 500 viruses, 1,000 viruses in one cell. If the virus is very efficient, maybe a dozen of those will actually get to infect another cell. Okay? But each one of those will make 1,000 more. Okay? And so very quickly you get sick. Okay? I mean, we've all had that feeling, right? You go to bed and you got the tickle in your throat, right? Oh, I think I might be coming down with something. And then the next day you wake up and you wish you were dead. Okay? Because you're so sick, okay, that it's just awful, right? And it's just happened overnight because a virus can reproduce that quickly. Okay? So that's essentially how a virus works, and vaccination works very similar to that. Okay? We're just trying to, so when you get certain types of vaccines, they're either a dead version of the virus, okay, in which case your body recognizes that protein as foreign, learns how to destroy it, and then your immune system remembers how to do that for later. So you've had something that can't hurt you in there, a dead virus, and your body's learned how to kill it. Okay? The other thing that can happen is with mRNA vaccines, okay, mRNA is injected in and your cell absorbs it, and it learns from that mRNA, which is basically a type of genetic material, it learns how to become immune to anything that would be carrying the protein. So that mRNA that gets injected into you is the code for making the virus coat, not the virus itself, just the coat, okay? The protein part, and again, your body learns how to destroy it, okay? And we're finding that that is the best way, in fact, to protect people against viral infections of all kinds. If there's like one good thing that's come out of COVID, it's the development of effective mRNA vaccines, because these could lead to like vaccinations for all types of viral diseases, HIV included. Okay, so it's kind of a big deal, okay, to have had all this money get pumped into the development of mRNA vaccines. Okay, so yeah, it's kind of a big deal, right? Because I know there's like some misinformation out there about mRNA vaccines alter your DNA, they turn you into a mutant. No, they don't. Okay, they certainly don't do that. Okay, they teach your cell basically, here's a viral code that you might encounter, and here's how to destroy it. Okay, and that can last longer and be more effective than a traditional type of vaccine. So like AstraZeneca, for example, is a type of vaccine that uses the dead virus. Okay, and it's been found to not be as effective as mRNA vaccines are. Yeah. Okay. Now, cell theory. First point of the cell theory, all living organisms are made up of cells. Again, the debate rages on viruses, whether that counts for them. They're obviously not made up of cells. Okay, but they can be single-celled organisms, like a paramecium, which we'll look at in the lab tomorrow, okay? Or they can be a single-celled organism like an actinopod, which lives in seawater and has a silicate shell. It's almost like made of glass, okay? And then a multicellular organism like a cactus, okay? But organisms are made of cells, all living organisms. Okay? So that's the first point. Just like the first point of the atomic theory. All matter is made of atoms. Well, all living things are made of cells. All right, second point of the cell theory, and this is probably the most important one. What is the role of the cell? The role of the cell is to carry out the basic functions of an organism. All right? That point is going to be a recurring theme throughout the unit, even when we're talking about multicellular organisms later. The role of the cell is to carry out the basic functions of the organism. So basic functions would include cellular respiration. That's how your cells break down sugar and release the energy. Okay? Photosynthesis, if it's a plant or a photosynthetic organism, that's how they make sugar from sunlight, carbon dioxide, and water. Okay? Making proteins, getting rid of waste products. Okay? Stuff like that. Those are all basic functions of the cell. Every cell has to be able to carry out those functions. All right? Everybody with me there? Okay, so that one will come up. It'll be a big part of our cell anatomy lab that we do next Monday. Okay, but this is something that I always tell people when the unit exam comes up, that's something you could work into a lot of different answers. Okay, because it's something that's just going to keep coming up. All right, and then the third point, new cells are not created. They come from the division of existing cells. All right, so there's no factory in your body that produces cells. Okay? Cells come from the division of existing cells. So if we trace you all the way back, okay, to the moment you were conceived, all right, so when sperm met egg, okay, that made a single cell called a zygote. Every cell in your body now is a descendant of that first cell. Okay? That's why in the Catholic Church we believe that life begins at the moment of conception. There's a scientific backing for that. Okay? All of the genetic potential for you is present the moment you're conceived. Okay? That's why the Catholic Church believes it. Okay? So if we're looking at any cell that's dividing, okay, that cell copies all of its genetic material and then splits in half. Okay, so each of the two cells has all the copies of the genetic material, okay, and then so on and so on. So when you were a single little zygote, okay, that zygote started dividing, and eventually became 4 cells, and 8, and 16, and 32, and 64, and it gets bigger and bigger. Okay, starts to change shape and become the little embryo that would later become a fetus and then be you. Okay, so all of the cells that you have are a descendant of one single cell, and that's still true. Okay, your body produces new cells all the time. Okay, can stuff go wrong with that process? The division process is absolutely not perfect. Okay, it can work pretty well, not flawlessly, but pretty well, pretty accurately most of the time. Okay, but mistakes can be made. If a mistake is bad enough, it can lead to fairly major problems depending on when and where it happens. If we're talking about in the formation of gametes, like an egg or a sperm, okay, and something goes wrong with the division process that produces those, then there can be genetic disorders that the resulting baby can have. Okay, like for example, Down syndrome, okay, is a result of trisomy 21. That means that somewhere along the way, when either the egg or the sperm, okay, was dividing, one of the copies of chromosome 21 didn't go to the other cell, and so one cell retained 2 copies of it, which it should not have. Okay, and then when the egg was fertilized, there were 3 copies of that chromosome. Okay, now you would normally think, well, more is okay. Like, how can having too much genetic material be bad? Well, okay, I mean, you look at the effects of Down syndrome on somebody, right? It's a pretty major disorder. Okay? And it can have pretty major health implications for someone with it. Okay? So stuff like that can happen. Some trisomies and genetic disorders like that, they result in a miscarriage, even like very early on in the process. Yeah, so that process is not perfect. Later on, when you're a fully developed organism, okay, certain things can cause that process to go wrong, and it can lead to what disease? Cancer. Yeah. Okay. What cancer is the rapid division, abnormal rapid division of cells. Okay. So if you have even just one cell that's damaged and dividing out of control, okay, it forms a tumor, which is a big mass of rapidly dividing cells. Right? So that's a problem with that division process. Okay? So then we obviously have to treat it by either like surgically removing it, and then usually things like chemotherapy. Okay, chemotherapy kills rapidly dividing cells, which is why when you're on chemo, you lose your hair, okay, because that's rapidly dividing cells that produce your hair. Okay, you become immune compromised. Okay, your immune system relies on being able to rapidly produce white blood cells and things like that. Okay, your stomach usually have like lots of nausea because the lining of your stomach is also a rapidly dividing cell. So there's all kinds of other issues that go along with that. Okay, so those are the 3 points of the cell theory. Everybody good with that? Okay, let's talk about the important people. Okay, first important person, the person to discover cells. Hooke. Hooke wasn't a biologist. Hooke was a physicist. Okay, and this is one of these other areas where physicists feel superior to all other types of scientists. Physics discovered the atom, which is the basis for chemistry. Most important tool in biology, built by a physics guy. Okay, so you're welcome, other sciences. Yeah. Okay, so what Hooke was doing is he was actually doing experiments with the refraction of light in lens systems. Okay, so he was building these devices with different pieces of curved glass and seeing what he could do with the path of light. And he found that, you know, if you arrange them in certain ways, you could magnify very distant objects. And if you arrange them a little bit differently, you could magnify really, really small objects that were really close. Okay, it all depended on where you place the lenses and stuff like that. And so Hooke was basically just playing around, okay, with lens systems, and he needed something to look at under his microscope. Okay, and so what he did is he took a piece of cork. Okay, I don't know, maybe he drank. He had lots of corks laying around, I don't know. Okay, but cork is made from a tree. Right, so what he did is he took the cork and he sliced it really, really thin and placed it under the microscope. Now we're not talking about an extremely powerful microscope here. This thing could magnify about 30 times. Right, that's about what we would see on the first scanning power lens of our microscopes tomorrow. They're 40x. Okay, so they're about a little bit more powerful than Hooke's first microscope. Okay, and he put the cork underneath there, and this is what he saw. All right, and he said, this thing's got a bunch of empty rooms in it. It looks like a prison full of cells. Empty little rooms. Okay, oddly enough, that name stuck. We know cells aren't empty rooms. Okay, now we know they're full of all kinds of things. They're actually very busy, active places. Right, but what he was looking at was a dead tree. All that was left was the cell wall of all the plant cells that made up that tree, and that's what he could see, was these empty rooms. They were empty when he looked at them because that tree was dead. Okay? So he discovered cells, okay? Built the first microscope, kind of an important guy. Okay? All right, now, once lens systems started becoming, better, people started developing all different kinds of microscopes. Thankfully, not all of them caught on, like the one Anton von Leeuwenhoek was using, okay, which was this paddle-shaped thing, okay, which you would basically have to kind of look through this tiny little magnifier, this thing right here, okay, or if you had a really good situation. You could have a bright source of light, and you could have the light go through the lens and project like onto like a small screen or something like that. Okay, again, just using sunlight or whatever, reflected sunlight. It was good enough to magnify quite a bit more if you knew what you were doing. Okay, but it was difficult to use. But Leeuwenhoek used it a lot, and he was the first person to notice that cells are not empty. Cells are alive. Cells are full of all kinds of things. He looked at blood cells, okay? He looked at all kinds of stuff that was swimming around in pond water, okay, or drinking water at that time. Okay, kind of made a big deal. It's like, maybe these are all the things that make people sick, cause like the plague stuff, dirty water. Okay, and sperm cells from bulls. Don't ask me how I got those. Okay, 'cause I don't want to know. But yeah, he was looking at all kinds of things, and he was the first person to view living cells. He was the first person to watch cells divide and understand how that process worked. Okay, things like that. So that was pretty important, understanding that cells are not empty, okay, not static. They're very dynamic. They're changing all the time. All right. Now, his work led to the work of these two people, Matthias Schleiden and Theodor Schwann, two Swedish scientists, okay, who developed the original cell theory. Okay, Anton von Leeuwenhoek had discovered that cells were in lots of places. They were in lots of organisms. Okay, their idea was that cells are the basic unit of life. They should be present in all living organisms, and that was their hypothesis. If cells are the basic unit of life, and tissue samples from as many organisms as we can find are viewed under the microscope, then all of those samples will contain cells. Okay? It's a simple hypothesis. Okay? It became the basis of the cell theory. Right? It was confirmed over and over and over again. All right? From there, obviously, they added on to that. That was just the first point of the cell theory. Okay? In watching and observing what cells do and how they behave, they realized that they carried out the basic functions of the organism. Okay? And they also observed cells dividing, okay, reproducing, okay, and knew that there was no place where cells were just produced. They always came from the division of existing cells. Right? So their contribution was that they actually took all of that information, all their observations, and came up with the cell theory. All right. Questions there? So, as equipment gets better, we're better able to observe cells, see more and more parts of them, okay, and understand exactly what it is they do within the body, okay, things that can go wrong, things that happen all the time, okay. But what we did notice was, and this is from like Schleiden and Schwann's work, was that cells of very different organisms still have more in common than they have different. Okay? If you look at a plant cell and an animal cell side by side, okay, I mean, other than the cell wall here in the plant cell and chloroplasts in the plant cell, there's really not a lot different. Okay? Animal cells have centrioles and plant cells don't need them. Okay? But really, there's a lot in common. And that's just a visual look. Okay? That's just appearance-wise. These are the structures they share. When you get down to the processes that a cell carries out, there's even more in common. Okay? Plants and animals use the same process for making proteins. They make the same process for breaking down sugar into energy. They use the same process to transport materials within the cell. All of that is the same. Okay? They have so much in common. And that's just plants and animals. That doesn't even include fungi, okay? Or like bacteria and protists and things like that. All of these organisms have cells and they all behave similarly and carry out similar processes. Okay? Which leads to this idea that they may have evolved, and likely did according to the fossil record, from a common ancestor. Right? Now, later on in the unit, just in case any of you are having trouble with me talking about evolution, okay, later on in the unit we're going to talk about how evolution and religion do not disagree. Okay? There's this big idea that people have that's very wrong, that the Catholic Church says that evolution didn't happen and that it's all wrong. That is absolutely not true. In fact, Pope Francis came out a few years ago to talk about that and say, no, we have zero problem with this idea and how it's, how it's supported by, passages from the Bible and stuff like that. And we'll look at all that. later on in the year. Okay, so just so you know, it's okay for me to say that. I'm not being a bad Catholic by saying it. And it doesn't make you a bad Catholic to believe in evolution either. Okay. Okay. All right. So microscopy. You know what? I'm going to give you guys about a 3-minute break here. I'm just going to go grab a microscope so I can show you some of this stuff. Okay? I'll give you a 3-minute break, and then we'll be right back to it. So, a couple things about the use of the microscope, because obviously we're going to be in the lab tomorrow using microscopes. All right? All the microscopes will come with these really cool dust covers. Some of them have the original dust covers, but not many. Okay? Most of them have been replaced by these giant freezer bags because, well, they keep the dust off just fine. And they're much cheaper than the actual dust cover. All right? So, you'll find that on there. Take it off. Microscope works much better without it on. Okay? Now, when you are carrying the microscope, okay, you need to carry it with 2, count them, 1, 2 hands. Okay? It is not to be carried like this. Okay? You carry it with 1 hand, you could drop it. Or, you see all the electrician's tape on here? Okay? What sometimes happens is people are not carrying with 2 hands and holding the cord, and the cord comes loose. And then it ends up on the floor. Eventually. There we go. Okay? It ends up on the floor. People don't really notice it. They're walking along and they step on it and they run out of cord. Or it slips right up to their foot and pulls the cord out of the charging block, or worse, out of the bottom of the microscope. And then I take the microscope apart, put the wires back in, and I am perturbed. Okay? So the other problem is that sometimes when that happens and we get to the end here, it pulls it right out of their hands, and then it falls to the ground. And we hear the crashing sound, followed by the ambulance sound. Don't drop the microscopes, please. Thank you. They're kind of expensive. Okay? We don't want to have them broken. All right. So always carry them with 2 hands. Okay? Microscopes are always returned with the lowest power lens in position. So if you're looking at this one, see how the shortest lens is in viewing position? That's what people expect to have in viewing position when they get the microscope. So just make sure that that's returned that way. Because sometimes people don't check, okay? And the last lens you're likely to use is the high power one, so the big one. Okay, so the worry is that if we leave this in position, somebody can actually damage both the slide as well as the microscope itself. Okay, so if they think that the low power or scanning power, lens is in position, okay, they may grab the coarse focus knob, that's the big one, and they can drive this thing right down into the slide if the brake hasn't been set properly. And sometimes these brakes get kind of pulled off. And like this one here, it hit the brake before it hit the slide. Okay? But some of them, those are off a little bit. Okay? They've slipped or whatever, and you can actually drive the microscope lens right into the slide, which will damage both of them. Okay? And that's a really expensive repair that I can't do. Right? So we need to make sure we're just careful about using that. Okay? So make sure you're putting the scanning power lens back in position, because that's the one you're going to start with as well. Okay? When using the microscope, always start with the lowest power. That's the scanning power lens. Move up to the low power. Move up to the high power. Okay, I'll talk about what you're looking for when we get there. Okay, and always use lens paper to clean the lenses. Okay, now there's special paper in there. It's just labeled lens paper. Okay, that's what you use to clean like this part and these lenses here. Okay, the reason for that is that there can be grit on, you know, your shirt or your finger or whatever, okay? And we don't want that grit to scratch the lens. Because even if you put a microscopic scratch in a 400x lens, okay, it goes from being a microscopic scratch to the naked eye to being the Grand Canyon when magnified 400 times. Okay, so we don't want to scratch the lenses, so we use the lens paper once. Okay, take the lens paper, wipe it, and throw it away. Don't use it a second time. Okay, but always use lens paper, because inevitably I see people and they're like, oh, like in COVID, really don't do that. That's gross at the best of times, but in COVID, really don't do that. Okay, you know, or they're like, and then they take their sleeve and they do this. Okay, there can be grit on your sleeve. Okay, and that can scratch the lens. Okay, so you have to really make sure you're using just the lens paper to take care of that. All right, a couple other things about these particular microscopes. Okay, the stage can be moved. Okay, there's knobs here to move the stage around. That's going to be really handy when you're trying to find stuff on scanning power, okay, and to get things centered in your field of view. What I mean by field of view is this. When you look through the microscope, you see a lit circle, right? Your specimen, let's just say it's right here, okay? I want that specimen to always be in the middle of my field of view. So what I do is I use the little knobs here, okay, the ones on the bottom, and that can move the stage around, rather than taking my finger and pushing on the stage and trying to move the slide around. Okay? There are some microscopes where you have to do that, but not ours. Right? They have this nice stage clip here, okay, that will hold the slide in place, okay, there on the stage, and then it'll move around with the stage knob, and that makes it way easier to move. Okay? But we always want our specimen to be centered so that when we move up to the next magnification, it's still going to be visible. Right? Because if I leave it out here, say it's right near the edge, okay, and this is my scanning power lens, when I move up to my low power lens, I don't see as much area because I'm now going to magnify even more. So there's what I see on the next magnification. Now I no longer see my specimen. Right? And when I move up to high power, I see an even smaller. More greatly magnified, obviously, okay, but an even smaller area. Right? It's kind of like this. Let's say you go to like a Flames game, right? You're sitting on one side of the arena, and your friend says, "Hey, I'm here too. I'm wearing like a bright orange shirt," for whatever reason. He's not wearing a Flames jersey, but okay, he's wearing a bright orange shirt. Okay, I'll wave to you. So you're like, okay, great. What's the best way to find him? To scan the crowd with your naked eye until you see an orange shirt that's waving, or to take out a pair of binoculars and look at each person individually? Scan it. Then, once you think you've got somebody in an orange shirt, grab the binoculars and check that one person. That's what we do with the microscope. I want to look at the scanning power first, okay, so I can see as much of the slide as possible, but very little magnification. Find my specimen, move it to the center, and then move up in magnification. Okay, everybody with me there? All right, so that's what we want to do. Okay, always move it to the center. How do you know it's in the center? Well, conveniently, all of the microscopes actually have this wire. Okay, and it'll look black. It's actually like brass, but it'll look black under the microscope. That goes to the middle. We call it the pointer. It's supposed to be there. Okay, I only say that because I've had the odd group who'll bring up like 4 microscopes. Mr. Kuda, there's something wrong with this one. Mr. Kuda, there's something wrong with this one, and I'm looking through and like, can you tell me what's wrong with it? Well, don't you see that big black thing? The pointer. Right. That's what that is. So I'm just telling you that's supposed to be there. It tells you where the center is, okay, just so you're not confused. Okay, but that's what we use it for. All right. I am never going to ask you to label the parts of the microscope. But I do need you to know what the parts of the microscope are, at least at a functional level, so when I'm talking about it, you know what I'm talking about. Right? So, the important parts of the microscope for us, okay? The eyepiece and ocular lens, okay? That's the part that you look through, okay? It rotates. That way you don't have to pick up the microscope so all of your group members can look at it, because every time you pick it up and set it down, it's probably going to go out of focus. Okay, that vibration of setting it down again will probably ruin your focus, and then you'll be setting it up again. So best to just have it in one place and move the eyepiece around, okay, so everyone can use it. Now, obviously tomorrow, when we're doing this, we're all going to be wearing masks. There is going to be a bit of an issue with fogging. Okay, it's not the end of the world, but you're going to have a bit of an issue with fogging. Okay, I would also recommend that if you wear prescription glasses and you have contacts, wear your contacts. It's way easier to look through a microscope with contact lenses than glasses. Now, if all you have is glasses, obviously you still do it that way. For me personally, I find it easier when I'm in my contacts rather than my glasses to look through the microscope. Okay, that'll probably change more as I get more and more farsighted, but okay, for now. Okay, so for you guys, I would suggest that. Okay? The other things we need to know about, okay, this is the arm here, okay, that's for carrying. One hand goes on the arm, one goes on the base, so this is the base. Okay, you've got your revolving nose piece where all the lenses are, the stage, okay, the stage clip that holds the lens in place, the stage adjustment knobs, and then you have your two focusing knobs, the coarse adjustment knob and the fine adjustment knob. Okay, the difference is how much they move the stage. Okay, if you watch me here, right, if I turn the coarse focus knob, it's not even a full turn before I've moved the stage through its full range of motion. Right? So it focuses very, very fast, which means you're only going to use it on the scanning power lens, maybe on the low power. It's useless on high power because you'll go right past focus so fast you won't even know what's happening. Okay? So your fine focus knob, that's what this is for. You can see I can turn it like crazy. It's geared differently. It hardly moves the stage at all, and I can just keep turning and turning and turning and turning, okay? And the stage moves very, very little. That's important when you're on high power. All right, other things, these have to do with lighting and the quality of your image. Okay, there are little adjustments underneath. Okay, there's the condenser diaphragm. That's this little tab that I'm moving right here. Okay, it opens an iris underneath. Okay, that lets more or less light through, which can help with the contrast. So if you're looking at something that's you can't see the edges of it very well, changing the amount of light that comes through can help that. You also have this rheostat here that can dim or brighten. Okay, the light that's coming through, you'll probably just want that on as bright as it'll go and then adjust from here. Okay, and that's really all you need on there. Okay. Yeah.
Do we have to wear like safety goggles?
You can, you don't have to. I've only had one person get a black eye doing this lab.
Do we want to know how?
No, they just,
Oh. Ow.
yeah, like it was the weirdest thing. I don't know how exactly they didn't, like when they were getting towards the microscope to look in it, like I don't know what happened, but they, bang, right there, you know, yeah, black eye, but it's only happened once, so I'm gonna, and they didn't lose their eye, so I'm gonna go with, unless you're really clumsy and know that about yourself and want to wear safety goggles, you don't have to wear safety goggles to use the microscope, okay? I mean, they're hard enough to see through and you don't have to see really fine things. All right, now, here's what I was talking about before, okay? And I'm going to need you to copy this down onto a piece of paper because this is going to help you with your size estimations, okay? Because when we do a lab diagram, we have to tell people the size of the objects that we're viewing. Okay, so as I was saying before, your scanning power lens is your lowest magnification but largest field of view. Okay, field of view means how big is the circle, okay, or how big of an area can we see through that particular lens. So when I'm looking through the scanning power lens, okay, I'm magnifying 40 times, and my field of view, the diameter, okay, is 4,000 micrometers. Right, to give you an idea, that's 4 millimeters. Okay, so the distance from one side of your field of view to the other is 4,000 micrometers. Now, this little symbol here is mu. It's the Greek letter M. Okay? Why do we use mu for micro and not M? Because we already used M for milli. Okay, so we can't use it again for micro, so we use mu M for micro. You can find that symbol, okay, if you go to like insert symbol in Google Docs, you can find it, or I will accept if you write a U. Okay, so you can use UM, or you can find this, the mu symbol and insert it. Okay, it's up to you. I'll accept either one. Okay. Just don't put millimeters, because if you tell me this amoeba is 4,000 millimeters, okay, you're telling me that you have a 4-meter amoeba, okay, which you do not have. All right, on our low power, okay, our next lens up, so the one with the yellow band on it, okay, it magnifies 100 times, and this diameter is 1600 micrometers. So I don't see as much area, but I've magnified what's there more. And then on high power, okay, my field of view shrinks to 400 micrometers because my magnification increases to 400 times. Okay, so what I'm looking at is in a very, very small area now, but it's 400 times larger than it appears to the naked eye. So you're probably wondering, so why is this stuff important? Here's why this stuff is important. Let's say I'm looking at something on high power. So I know that the distance from here to here is 400 micrometers. Okay? Let's say that what I'm looking at looks like this. Approximately how big is that? I'd say, yeah, 200 would probably be on the upper end. I was thinking more like 150. Yeah, okay, it's a little less than half, right? But I mean, it's an estimation, right? When we are estimating the size of an object, that is exactly what we are doing. We are estimating its size. We are not calculating its size, okay? We're doing our best to give an approximate size of that object. I would probably, for something like this, accept anywhere between 100 and 200 micrometers as an acceptable size for that, as an estimation, right? So that's all I do. I know that distance is 400. All right, it's a little less than half of it, so I'll say it's this much. That's all you have to do. You don't have to calculate, you just have to estimate the size of the object. When we do the size estimation is done along the longest dimension of the object. Okay? All right. Questions so far?
When you're estimating, do you always estimate in the high power?
It depends what your end's magnification is. Tomorrow, the first thing we're going to look at is the letter E. We're not going to look at that at anything other than scanning power, because after scanning power, you can't see the whole E anymore. So you'll do this size estimation on the scanning lens, and it'll be, you know, thousands of micrometers as opposed to 150. Okay, other questions there? Okay, so that's the numbers there. Okay, we talked about those already. Okay, estimating the size of an object, we talked about that as well. Okay, so it's, like I said, it's just an estimate. It's just an estimate. All right. Now, in terms of lab diagrams, we're going to talk about how we're going to do that tomorrow in a bit more detail, and we'll look at a bit of the format today. Okay, but what you're going to do to make a lab diagram is you're going to use your phone to take a picture through the microscope. Okay, the first couple times it's going to be tricky. Okay, because you've got to hold that phone just right for the light to come through the lens of the microscope and into the lens of your camera. Right, so you're gonna have to get it just right. Okay, and then you can, you know, snap, get it focused, right? Do all that stuff. You guys all know how to take good pictures with your phone, right? That's what you're gonna do. You're gonna take a picture with your phone, and then you're going to insert that into a Google drawing, which will then become part of a lab drawing in your lab report. Okay, and I'm going to show you how to do all of that tomorrow. It's really not that hard. Okay, just make sure you get as good a picture as possible. You know, really hold it still. You might even need, you know, someone to hold it and someone else to do the, you know, the pinching and the focusing and stuff like that, because you want to get a good picture, okay, through the microscope. And it'll take a little bit of practice, right? So I think what we'll do tomorrow is we'll forgo having a quiz. I know that's going to break your hearts. Okay, but we'll forgo having a quiz so we get as much time as possible to go over lab drawings, to you know, get in there and use the microscope and get the hang of how to use it. Okay, have lots of time to get good pictures and all of that stuff. Okay. All right. So in terms of lab drawing formats, okay. So a lab drawing okay, is a bit different than what you might think, okay? And that would mean that, oddly enough, the title doesn't go at the top. The title goes below. Okay? So you're going to have your specimen on a piece of paper, okay, and you're going to make sure, or well, in this case, in a Google Doc, okay, and you're going to have it take up as much of a single page as possible, right? Okay, so let's say that this is one page in your Google Doc, okay? The diagram, the actual picture, should take up as much of that single page as possible. You cannot get more than one lab diagram on a single page, right? That's how big it should be. So if I'm drawing, you know, like an amoeba, which is kind of just a randomly shaped organism, like that, okay? That's how big I want the picture to be. Make it as big as possible, right? Your name and the date go in the top right. The title, the size, and the magnification all go underneath. And your labels all go to one side. Okay? We don't want this amoeba to look like it's exploding. Okay? Because that's what some people do. They put the picture right in the middle and they got labels going everywhere. Okay? And it looks like the amoeba or whatever it is you're drawing, it looks like it's exploding and it looks very disorganized. Okay? So what we want is we want all of our labels to go to one side and we want them to be straight lines. Except that one's not straight, but now it is. All right, so all of the labels go off to the same side so that all the labels are right there and easy to see. Okay, if you've got two structures that are kind of right beside each other, so let's say I was labeling like, you know, this structure here, and there was another structure kind of over here that I needed to label, right? Then I do this. Diagonal line up and then over. Everything ends in a horizontal line. Okay. You're probably thinking, wow, that's really picky. I completely agree with you. This is the format of a proper lab drawing. I can't help that it's, yeah, picky. Okay? But I'm trying to save you some pain later. Because you lose a lot of marks for format in university if they're not right. All right, everybody good with that? I'll show you the nuts and bolts of how to do that in a Google Doc and in Google Draw tomorrow before we go across the hall to the lab. I'll show you that.
So we're just drawing the picture, or
No, your picture is gonna be inserted here.
Oh.
Your photograph will be here, and then you're gonna just put the lines on and all the other stuff on them. Okay? Because that's way better than drawing. I always, like in university, I had to draw by hand with a pencil, shade and all that, and I'm a terrible, terrible artist. Okay? And so I always, I would have loved if digital cameras had been a really big thing then, that would have been so much better. Like you guys are very lucky that you can now do it this way. You can take good pictures of things very easily, and that'll be much, much easier for you. All right, so guys, we're just starting this unit today. This unit will take us to middle of November, probably like just before parent-teacher interviews or so. We're a little bit behind. We took a little longer in chemistry than I normally do. So we're just a tad bit behind, but we should finish this up right around parent-teacher interview time. We'll do physics, okay, for all the way up to Christmas. Hopefully we finish it before Christmas, but if we don't, we'll have to finish it after, right? And then our last unit, I think is going to be pretty short, pretty rushed. It's less important than the other three. Okay, but that's where we're sitting. Okay, global systems and energy. Yeah, it's kind of a hodgepodge of everything. So. All right, so that's what's coming up for you guys. So obviously you'll have this lab. You'll have another lab on Monday of next week. Okay, we'll have a few in-class activities. Those will be ones that you do in the class period and submit at the end of the period. They're not designed to be homework. They're designed to be done in a class period. Okay, there'll be a few of those in this unit as well, and one research thing on a plant of your choice. When we get to plants, you can start thinking about that. But yeah, that's where we're sitting. Don't forget, if you need some help with anything, and lots of times you can come and see me. Know that for biology, the way that you prepare and the way that you study is very different from what you did in chemistry. Okay? In chemistry, it was all about practicing your skills and learning how to do certain things and stuff like that. In biology, it's really about reading your notes, picking out what's important, rereading that part, memorizing some things, labeling, right? Oftentimes, like cue cards or recipe cards are really good for this unit, like making up, little flashcards for yourself. That can be really useful for biology because there's a lot of memorization. So if you're looking for ways to start preparing and reviewing and stuff for bio, those are your better strategies. All right. Okay. Questions from you guys? All right. Then we're done for today.